4 mineralized tissues are found in the oral cavity. 3 of these, enamel, dentin, and cementum, are components of the tooth. Today, we're going to talk about the least mineralized one, cementum, a hard, avascular connective tissue that covers the roots of teeth. The development of cementum can be subdivided into 2 main stages: a prefunctional stage, which occurs during root formation in the intrauterine life and primarily covers the cervical third of the root. A functional stage, which begins once the tooth is in occlusion and continues throughout life, covering the apical 2/3. Although cementum formation takes place along the entire root, its initiation is limited to the advancing root edge. At this site, Hertwig's epithelial root sheath derived from the coronoapical extension of the inner and outer enamel epithelium send inductive signals to start cementum formation. The precursor cells of cementoblasts and periodontal ligament fibroblasts reside in the dental follicle, a sac-like connective tissue structure surrounding the developing tooth germ, and it contains precursor cells that give rise to cementoblasts, cementoblasts to form cementum, or fibroblasts to form the periodontal ligament, or osteoblasts to form the alveolar bone. We can divide the cementogenesis into 5 stages. 1. Initiation. As the root begins to elongate, the Hertwig's epithelial root sheath extends apically, make contacts with the dental follicle cells, and induces ectomesenchymal cells in the follicle to differentiate into cementoblast precursors through molecular signaling. 2. Disintegration of Hertwig's epithelial root sheath. For cementum to form, HERs must fragment, allowing direct contact between follicular cells and the unmineralized root dentin, or the mantle dentin. This matrix contains collagen-type iodine, DSPP, and bone sialoprotein, all critical elements for cementoblast attachment. 3. Differentiation into cementoblasts. Once exposed to root dentin, cementoblast precursors fully differentiate. These cells become cuboidal or polygonal, with large oval nuclei, and align themselves along the dentin surface. 4. Cementoid secretion. Cementoblasts begin secreting cementoid, the unmineralized organic matrix of cementum. It's deposited in thin layers and contains collagen type 1 and non-collagenous proteins like osteopontin, bone sialoprotein, and cementum attachment protein 5. Mineralization. Over time, cementoid becomes mineralized through the deposition of hydroxyapatite crystals, reaching 45 to 50% mineral content. This makes cementum the least mineralized of the 4 oral hard tissues, after enamel, dentin, and bone. This stage begins after the tooth erupts and comes into occlusion, meaning it contacts its opposing tooth. Unlike the prefunctional stage, this one continues throughout life. Secondary cementum is formed in response to external stimuli, such as mechanical stress, tension in the periodontal ligament, signaling, molecules that guide tissue remodeling. Its purpose is to help the tooth adapt to functional demands, like occlusal pressure or slight movement. For example, during passive eruption, the tooth moves slightly in an apical direction. To maintain its position within the alveolar socket, cementum formation increases at the apex, filling the space and anchoring the tooth securely. That's why the apical portion of the root typically shows thicker and more developed cementum than the cervical part.